Key Multiplicity Issues in Clinical Trials, Part 2. Introduction. Welcome to the second part of my online training course on Key Multiplicity Issues in Clinical Trials. Part 1 is available on this website, and its main goal was to introduce key concepts and approaches to handling multiplicity in what we called a traditional setting. Traditional problems, considered in Part 1, feature a single multiplicity component, or in other words, a single source of multiplicity. And in Part 2, we are ready to tackle much more challenging and consequently more interesting problems arising in confirmatory trials with several sources of multiplicity. The last 15 to 20 years have witnessed a surge of statistical methods for addressing complex multiplicity issues in clinical trials. This increasing interest reflects important trends that we observe in clinical drug development. Complex sets of objectives are introduced to improve the efficiency of clinical trials or entire development programs, and it's very common to come across phase 3 trials that rely on combinations of clinical objectives based on the analysis of multiple clinical endpoints, multiple doses or regimens of experimental treatments, multiple patient populations, and increasingly multiple decision points, which includes interim and final analyses. Evaluation of these multivariate objectives gives rise to increasingly more complex multiplicity problems and leads to inflated false positive rates. 2 regulatory guidance documents have been released recently to discuss the impact of multiplicity in confirmatory trials and statistical methods for handling multiplicity. The U.S. Food and Drug Administration, FDA, published the draft guidance on multiplicity issues in clinical trials in January of 2017, and the European Medicines Agency, EMA, released a guideline entitled Draft Guideline on Multiplicity Issues in Clinical Trials. This guideline is a revision of a points to consider document that was published back in 2002. These draft guidance documents on multiplicity issues in clinical trials provide information on traditional settings that we considered in Part 1 and advanced multiplicity problems that will be the main focus of this course. I was very pleased to find detailed discussions of commonly used multiplicity adjustments in advanced settings, for example, in clinical trials with hierarchically ordered endpoints. This includes different types of gatekeeping strategies. So I have included multiple references to both guidance documents in this course, and we will get to discuss how the guidance documents approach some of these challenging multiplicity issues. By way of outline, let me reiterate that in Part 1 of this course, we focused on traditional multiplicity problems with a single source of multiplicity, or in other words, problems with a single multiplicity component. This online training course, Part 2, deals with advanced multiplicity problems, or multivariate multiplicity problems with several factors, each defining an independent source of multiplicity. The more straightforward traditional multiplicity settings can be visualized using this simple diagram. In univariate multiplicity problems, we define a single family of null hypotheses of no effect. They represent the same source of multiplicity. Examples of settings with a single source of multiplicity include clinical trials with multiple endpoints, but a single dose or regimen of the experimental treatment, or on the other hand, it could be a single primary endpoint evaluated at several doses. It will be helpful to very quickly go through the topics covered in Part 1. We started with a description of clinical trial examples. We spent a good amount of time on defining key concepts that play a central role in traditional as well as advanced multiplicity problems, including inferential goals, commonly used definitions of error rate, classification of multiple testing procedures. We also went over popular multiple testing procedures, beginning with non-parametric and semi-parametric procedures that rely on a data-driven hypothesis ordering. We also discussed non-parametric multiple testing procedures that are based on a predefined hypothesis ordering. We introduced commonly used classes of parametric multiple testing procedures. We defined simultaneous or multiplicity adjusted confidence intervals, and finally discussed a very important topic of power calculations that rely on simulations in the context of clinical trials with complex clinical objectives. In part 2, as I have explained, our main focus will be a broad class of problems where we need to account for multiple sources of multiplicity. To accomplish this, clinical trial sponsors typically group the individual null hypotheses of no effect into several families, as shown on the slide. The families are typically tested sequentially, beginning with the most important family, family 1 in this diagram. Each family in the sequence, except for the last family of course, serves as a gatekeeper for the next family, meaning that certain conditions must be met within the current family to pass the gatekeeper and move on to the next family of hypotheses. Two examples of settings with several sources of multiplicity are given on this slide. First of all, it is very common to see clinical trials that employ multiple endpoints and multiple dose control comparisons. The analysis of multiple endpoints, which includes primary and secondary endpoints, defines the first source of multiplicity, and in addition, multiplicity is induced here by the analysis of the endpoints at different dose levels. The second example deals with multiple endpoints that are analyzed in multiple patient populations. Once again, the first source of multiplicity here is the analysis of several clinical endpoints, and in addition to this, we have a set of analyses that focus on evaluating the treatment effect within all of the predefined patient populations. Those analyses define the second source of multiplicity. We are now ready to go over the modules included in this course. Module A, Clinical Trial Examples. The main goal of this module is to define clinical trial examples that will be used throughout this course. These examples will be used to help motivate commonly used methods for tackling complex multiplicity problems, and I'm also going to use the same examples to illustrate various testing strategies in the individual modules. Module B, Introduction to Gatekeeping Strategies. We will define a general framework for setting up testing strategies in clinical trials with several sources of multiplicity. They are known as gatekeeping strategies. I will introduce commonly used classification schemes that will help us understand the structure of popular gatekeeping strategies. We will also review relevant regulatory considerations in confirmatory trials with complex objectives using the recently published FDA and EMA guidelines. Module C, Parallel Gatekeeping Strategies. This section will focus on a popular class of multifamily problems where each family serves as a parallel gatekeeper. We will introduce simple gatekeeping strategies based on Bonferroni type procedures, for example on chain procedures, also known as graphical methods, and then more powerful strategies that are derived from semi-parametric procedures, for example Hogberg-based or Hummel-based gatekeeping procedures will be presented. Module D, General Gatekeeping Strategies. Very general multiplicity problems with arbitrarily complex structure will be considered in this section. We will refer to testing strategies used in this setting as general gatekeeping procedures. As in Module C, we will concentrate here on efficient strategies based on semi-parametric gatekeeping procedures that are more powerful than basic Bonferroni-based gatekeeping procedures. Module E, group sequential trials with multiple objectives, and Module F, general adaptive trials with multiple objectives. These two modules deal with complex multiplicity problems with an additional twist that the treatment effect will be evaluated at one or more interim analyses in addition to the final analysis performed at the end of the trial. We will begin with group sequential trials with several multiplicity components, and we will then consider multiplicity issues in the context of general adaptive trials. This is a new and exciting topic. I did not cover adaptive designs in part 1. And finally, Module G, power calculations. Simulation-based approaches to power calculations in clinical trials with multiple objectives will be discussed in Module G because we can no longer rely on analytical approaches in these complex settings. The Mediana package that supports simulation-based power and sample size calculations in a very broad setting will be used in this module to perform power calculations in trials with multivariate multiplicity problems. Now I would like to introduce two books that deal with the general topic of multiplicity in clinical trials. First, Analysis of Clinical Trials Using SAS, edited by Professor Gary Cook and myself. The first edition of this book was published by SAS Press in 2005. We have updated the book, and the second edition was published in 2017. The second edition covers a broad range of statistical topics from early phase to late phase trials, and Chapter 5 gives an introduction to multiplicity issues arising in clinical drug development with a large number of case studies based on phase 3 clinical trials. The chapter begins with traditional multiplicity settings, but in the last section of the chapter, we focus on advanced multiplicity problems with several sources of multiplicity and introduce popular gatekeeping strategies, and also touch on many important topics that will be presented in this course. The second book is Multiple Testing Problems in Pharmaceutical Statistics. This book was published in a Chapman and Hall CRC Press series in 2009. It was edited by myself, Professor Tamhani, and Dr. Bretz. The book provides a comprehensive treatment of multiplicity problems across the development cycle, from a preclinical setting to a clinical trial setting. There are 7 chapters in the book, and each chapter gives a detailed overview of methodological issues of a certain class of multiplicity problems or multiple testing procedures. There are multiple case studies, discussion of regulatory considerations, review of software tools to implement multiplicity adjustments. One of the chapters, Chapter 5, focuses on challenging problems arising in trials with several multiplicity components. Finally, as I did in Part 1, I would like to point out that you will find information on recent developments in the area of multiple comparisons in clinical trials, in a number of review papers and tutorials in biostatistics that were published in Statistics in Medicine. These are the papers on key multiplicity issues in drug development, and also 2 tutorials, traditional multiplicity adjustment methods in clinical trials and advanced multiplicity adjustment methods in clinical trials. You will find supplementary materials for this course on the Instant Training website, sprmm.com. On the main page of this course, there's a section entitled Supplementary Materials, and you can download the entire slide deck and also a link to SAS and R code from the software implementation sections. And you'll also find a list of all references from the individual modules.